So, in continuation of my last lecture, I would like to tell you that in case of solid state fermentation, we have already seen that in that particular fermentation medium, no free flow liquid was present. Now, here when I had discussed about this fermentation, fermentation process, I have told that the fermentation can be divided into two major groups. One is the submerged fermentation, another is the solid state fermentation. Now, when we are talking about the modified solid state fermentation, then we have done some modification to the traditional fermentation process. That means, while yesterday when I was discussing about the solid state fermentation, I have already mentioned you that heat generation is one of the major problem in case of solid state fermentation. To overcome that particular problem, what the scientists they have done? They have modified the reactor design in such a way that instead of putting it inside the tray, what I have told you that perforated trays are needed to dissipate the heat generation. Now, in modified solid state fermentation, what is done? The reactor vessel is constructed in such a way that in this is a solid base pair, and to which some sheaves are kept. Now, when it is kept, this sheaves has got some pore size, definite pore size, and or maybe the cotton wool that sheaves are there, to which the solids are kept. In the bottom, what it is done? The liquid nutrient medium is kept. Now, when it is kept, when it is the liquid medium is there, this liquid medium has got a constant touch with this porous bed, to which the solid substrate is kept. Now, during fermentation, when heat generation is taking place within this bed volume, as this particular bottom side is continuously in touch with this liquid medium, continuously liquid is coming in contact with this bed, and heat is coming and getting absorbed to this bottom liquid, and so the heat generation within the bed is taken care of. It is controlled by the liquid which is there present in the bottom of this reactor, and here it has been seen that with this type of modifications, the productivity or the yield of a certain targeted product is enhanced many-fold, and that is the reason why it is called the modified solid state fermentation. Now, in case of yesterday, what I have told, that in the tray type of reactor, this is the perforated tray which is present, and then we used to do traditionally the aeration, air passing, air circulation was done. So, instead of doing that, this modification has been done to this tray type of reactor, and that is called the modified solid state fermentation. Now, here also some problems are there, that is a handling problem. That problem of automation of this type of system. So, it is considered that though there is no thumb rule, there is no definite processes which is the definite what to tell, that there is no such the definite rules for selection of any biological or any microbiological processes. But if we are handling any filamentous fungi, then we are generally selecting the solid state or modified solid state fermenter. And if it is a bacterial system in general, then we are going for the submerged fermentation, as because filamentous fungus is a thread like structure, and yesterday I have told you the problems associated with that type of organism handling. And as bacteria is unicellular in nature, and bacteria has got its cell wall and cell membrane within it, and it is very easy to handle to produce the metabolite in the submerged type of fermentation. Now, when we are going for this submerged fermentation, then we can classify this submerged fermentation into the batch process, fed batch process, and the continuous process. Now, what is the batch process? This batch process is that in this particular system, all ingredients used in this bioreaction are fed to the processing vessel at the beginning of the operation, and no addition and withdrawal of materials take place during the entire batch fermentation. That means, whatever material we are just keeping after the run is over, fermentation is over, the entire batch is getting dismantled and entire volume of liquid is taken out of this system, and this is the batch process. While coming to this continuous process, what we are doing in this process, an open system is set up. Nutrient solution is added to the bioreactor continuously, and an equivalent amount of converted nutrient solution with microorganism is simultaneously taken out of the system. That means, if nutrient this nutrient is continuously coming, and continuously your product or the converted that after biotransformation, after fermentation, the fermented broth is continuously going out of this system. The inflow rate and outflow rate is at a constant level, so that the entire volume is maintained at a constant level. And here the flow rate is in such a way that the nutrient is getting the entire time of fermentation, conversion time, that nutrient they are getting to convert to the desired product. In case of fed batch process of fermentation, substrate is added according to the predetermined feeding profile as the fermentation progress, and this is the fed batch type of fermentation system. Now, if we see the continuous fermentation process, the continuous fermentation process can once again be divided as without feedback control and with feedback control. Now, when we are coming to this without feedback control, we are mainly considering the chemostatic model. Now, here with the chemostatic model, when we are considering this, the feed medium is containing all the nutrients is continuously fed at a constant rate, and the culture broth is simultaneously getting removed from the fermenter at the same time. That what I have told you, the feeding rate, when the the nutrient is coming inside this reactor, and when it is going out, the flow rate of this inlet and outlet is maintained at a constant level, so that the entire volume is getting maintained. Now, when we are coming to this with feedback control, it can be divided into three major groups. One is called turbidostat, another is the nutristat, another is the foxystat. Now, coming to this turbidostat, it is a continuous process to maintain the cell concentration at a constant level by controlling the medium, the feeding rate of the medium. So, this is the turbidostat. When we are coming to the nutristat, it is the cultivation technique to maintain the nutrient concentration at a constant level. Now, what is happening when we are talking about the different type of fermentation process? In case of batch process, what is there? When we are just taking any medium and we are inoculating with the live cells, then fermentation, and we are just incubating, then fermentation starts. Now, when fermentation starts, we are just expecting that after a controlled time of fermentation, we will be getting our desired product. Now, in reality, inside the reactor, what is happening? Now, when we are giving this carbon, nitrogen, all these basic nutrients which are needed for this particular biological product formation, then it is in the maximum that at 0 minute that it is the nutrient carbon content of this media is 100%. Now, when it is that inoculation is taking place, now when we are just inoculating this particular conical with some inoculum, this inocular, that live cells are coming and they start their normal activities. When they will start their normal activities, they will start consuming the carbon and nitrogen for their cell maintenance, for their growth, for their metabolic activities. So, what is happening when these metabolic activities of the cell started inside this reactor system, the nutrient carbon and nitrogen and other minerals which are there inside this reactor is continuously being uptaken by this microorganism. As a result, what is happening? The number of bacteria or number of cells or biomass concentration is gradually increasing, and carbon nitrogen content of that medium is gradually getting depleted. So, if we plot both these activities, then how we can plot that this way the biomass growth is taking place. This is the biomass growth, because media is very rich in nutrient, and when we are considering the carbon content of this media measuring, we find that carbon content is continuously getting depleted. So, this is the carbon, that nutrient content. Now, after after a certain time period, what is happening when carbon concentration is gradually getting depleted? These microbial cells which are grown and present inside the reactor is not getting sufficient nutrient, and if this is the biomass and this is the carbon content. So, if this is the graph, then we can find that after a certain time there is a crisis of nutrient inside the media. Now, in case of Nutristat, it is a cultivation technique to maintain the nutrient concentration at a constant level inside the reactor, so that there will not be any crisis of the nutrient and cells will be proliferating, cells will be doing their normal activities. In case of Foxostat, what is happening? It is the extended nutrient which maintain the pH value of the medium in the fermenter at a preset value. There should not be any change in this particular pH when the cells start proliferating, and these are some of these continuous fermentation process. Coming to the fed batch fermentation process, it is also without feedback control and with feedback control. When we are going for this feedback control, we can see that it is intermittent fed batch, it is constant rate fed batch, exponentially fed batch and optimized fed batch. When we are further classifying to this with feedback control, then indirect control and direct control. When we are going for the direct control, then we can go for the set point control, that is constant value control, and program control and optimal control. So, these are some of the technique which I think you have already learned in the bioreactor design system. Now, here this particular reactors with all control systems are used for the manufacture of biologicals from any living cells, irrespective of whether it is plant, animal or microbial. Now, when we are going for all control system, the sensor is playing a significant role to sense a particular parameter. Now, here in this particular, if we are coming to this sensor, then we can divide it into physical sensor, chemical sensor and biochemical sensor. Under this physical sensor, we can go for this temperature sensor, pressure, shaft speed, heat transfer rate, heat production rate, foam generation, gas flow rate, liquid flow rate, broth volume or weight, turbidity, rheology or viscosity sensors which are there, and these are the parameters which is influencing the product generation can be sensed in with this automated reactor system. When we are going for the chemical sensor, we can measure the pH just as I have discussed about the Foxostat. The pH should be maintained throughout the reactor, throughout the reaction time constant. So, when we are going for such type of fermentation, that means there should not be any change in the pH. So, with the sensor, that when it is we are sensing that there is a drop or increase in the pH, accordingly the acid or alkali or buffer system, whatever may be, is kept, is added drop wise to maintain the pH of that fermentation system. Now, here besides this pH, ionic strength, gaseous oxygen concentration, gaseous carbon dioxide concentration, dissolved oxygen, dissolved carbon dioxide, carbon sources concentration, nitrogen sources concentration, metabolic product concentration, minor metal concentration and nutrient concentrations are playing a significant role as far as this fermentation in the submerged and with controlled system is concerned. When we are going for the biochemical sensor, we are talking about the viable cell concentration. This is one of the very important parameter as far as the biological product is concerned. Now, here suppose we have inoculated our reactor system with the inoculum, but we do not know what is the percentage of living cell and the dead cell. If the percentage of dead cell is present there, that is the waste. It does not have any role in the product formation. So, here in some sensor are there through which we can go for the viable cell number or the concentration in the reactor system. We can also sense the NAD, NADH level of some of these particular cells. ATP, ADP, AMP levels are also can be evaluated through the sensor. Enzyme activity and broth composition can also get sensed with the sensor, which are already available and used in the fermentation system. Now, please note it that whatever I am talking about, this particular system are all aerobic process. Now, when we are talking about the bioreactor, then bioreactor is a vessel in which the chemical process is carried out that involve the organisms or biochemically active substances derived from such organisms. So, when we are going for that type of reactor selection, then reactor with internal mechanical agitations, that is impeller or turbine type of things are there. Sometimes bubble columns, that is which rely on the gas to sparge for agitation, and sometimes we are looking for the loop reactor, where we can tell that this mixing of the liquid circulation are achieved by the motion of an injected gas by a mechanical pump or by a combination of the two. So, these are some of the configuration of the different types of reactor, and I think you have already learned those things in some other classes where you have gone for the engineering aspect of this particular reactor system. Here today I will be discussing the biological part of this, means how these living cells are growing and what is the importance of each type of reactor as far as the living cells are concerned. Now, here when we are talking about the bioreactor, we mainly talked about the star tank reactor or bubble column reactor or some other type. So, here the star tank reactor, if we see here, the main objective is that aeration is there and we are just putting some impeller, and that for uniform distribution of oxygen throughout this medium, we are just using this impeller to rotate, so that uniform mixing will be there. Uniform mixing not only for oxygen, but for nutrient and other parameters which are associated with this particular reaction type. Now, when we are talking about the bubble column reactor, now here this particular reactor is little different from that type of this CSTR, that continuous star tank reactor, where here no impeller is there, and here what is happening, this air is coming and air is just purging inside this reactor, and through which the mixing is taking place inside the reactor, which is satisfying the oxygen demand and the uniform distribution of the nutrient throughout the medium. Now, mechanically star tank reactor with Ruston turbine agitators are highly flexible. It gives adequate oxygen transfer to the cell in water like medium. Now, here if the medium which we are using is just like water, that means viscosity, density, etc. are playing a significant role as far as the cell growth and the product formation is concerned. Now, let me give you one example. Say we are interested to produce gellan gum. Now, when we are going for that type of product formation, what we are doing? We are inoculating the bacteria to that particular environment, and gradually we can find that with the synthesis of this gum, this is a particular polysaccharide which is coming extracellular, which is coming out secreted by the microorganism to the surrounding environment, and when this gum is being secreted through the wall of this bacteria to this surrounding environment, that liquid, the viscosity of that liquid is gradually getting changed, and after a certain time it is so viscous that we have to stop and we have to take those gum out of this particular reaction type. Now, when such type of product formation is there, obviously we have to take the extra precaution, we have to have the idea about that how to purge oxygen, how to purge air, how to satisfy the organism to sufficient nutrient supply and all. So, here this type of reactor product formation, this selection of reactor system is playing a significant role. Now, when we are talking about such type of reactor, or say if we are going for the high cell density cultivation. Now, when we are going for the high cell density cultivation, what we are talking about, that the cell concentration in normal cases, if suppose we are getting 10 milligram per liter, but in case of high cell density cultivation, the cell concentration in the reactor is very manifold. It may go up to 110 milligram per liter. So, how manifold increase in that particular bacterial concentration? You just imagine. So, obviously, the viscosity of the nutrient medium which is there is not same to that of this initial. So, when such type of product formation is there, we have to achieve the oxygen supply demand, that the demand of oxygen for the microbes for that type of product formation, and for this sometimes with the air we have to purge the oxygen to satisfy that particular oxygen demand. And when such type of product formation is there, obviously the resultant broth what is there inside the reactor is totally having the different characteristics than what it was initially. So, if we are talking about these problems, then this high cell density culture which requires large oxygen transfer rate to support the respiring of the cells cannot be getting satisfied with this type of reactor system. Highly viscous broth where turbulence and hence high mass transfer rates are difficult to achieve, we should not use this type of reactor system. Now, there are some other reactor system where most commonly those bioreactors are highly flexible in nature and which has got mechanical agitator that disperse the gas bubble throughout the reactor, increases the residence time of the bubble, shears large bubble to form the smaller bubble. It provides the high KLa value. Now, when this KLa is the mass transfer coefficient, and here when we are getting the high mass transfer coefficient, obviously we can think of that, yes, this is one of the efficient bioreactor system, and where this at a constant airflow, if mass transfer level is getting increased. So, air is supplied by the sparger in such type of reactor system. Mixing is accomplished using disc or turbine type impeller, and baffles also augment the mixing of such type of things, and that is one of these reactor system, as I have also earlier discussed that it is a bubble column reactor. Now, here the dispersed gas bubble through the fermenter, which is using perforated plate to enhance the gas dispersion and mixing. It provides a low shear environment, although cells often accumulate at the surface of the bubble, and bubble bursting can damage or destroy the cells. So, these are some of these problems associated with this, but here if the cells are very fragile cells, then under no circumstances we can use the agitator. So, how to satisfy this oxygen demand of these cells? So, this is one of the technique. Through purging the air, we are just satisfying the oxygen demand of these cells. Energy efficiency, that is low power input required to transfer a given amount of oxygen relative to the stir tank reactors. Absence of mechanical agitation reduces the cost and eliminates one potential entry point for the contaminants. So, these are some of these this positive points and the negative points of bubble column reactor. So, how to overcome the problems associated with the bubble column reactor? So, the alternative to this bubble column reactor is the loop type of reactor. This loop type of reactor can be the airlift loop or propeller loop reactor or jet loop reactor. Now, see here this mechanism is same to that of the bubble column reactor. There also we purge the gas, and gas was just pushing this liquid out, and it was just getting mixed. In case of earlier, see this gas is just pushing this liquid, and liquid is getting mixed, and dissolved oxygen is coming in contact with this liquid, and oxygen, that liquid is getting dissolved oxygen, this oxygen is getting dissolved, and microorganism is taking that dissolved oxygen. Here you see inside one drop tube is there, and through which this, when this liquid is this purge, that air is getting passing inside this drop tube, and see it is pushing the liquid, and it has got one riser and one downcomer, and see from this drop tube this liquid is coming out of this the downcomer, and once again it is getting circulated. So, obviously this type of design has got little more advantages than the earlier reactor type. So, here obviously we can get the little better mixing efficiency, that the dissolved oxygen, that KLa value is little improved than the earlier one. Here see, here this inside drop tube, this liquid, this gas, oxygen, air is coming and it is pushing. This here you see, not inside the drop tube, from the outside air is going and it is just pushing the liquid, and through the drop tube once again it is getting circulated. Here through the nozzle that air is coming and it is just pushing this liquid, and this mixing the air with this liquid, coming in contact with this liquid, and it is just coming and getting it mixed. So, these are the different concept of airlift fermenter. So, it is improved version of bubble column reactor. Drop tube gives better circulation of fluid and mixing than the bubble column. It can give high mass transfer and relatively low power consumption, what we have already discussed in this particular reactor system. Now, when we are going for the any product generation, any product formation, the most popular fermented product is the ethanol. Ethanol is one of the most, it is the biggest fermentation process all over the world. Now, when we are going for this ethanol fermentation, now we mainly concentrate, we are mainly talking about the edible ethanol, but nowadays ethanol has got huge demand as far as biofuel is concerned, and that is the reason ethanol can be produced from different sources. One source is the that from the edible, another is the non-edible or the lignocellulosic sources. Now, when we are producing ethanol from sugarcane, so that process is one time, and because we are getting directly the sucrose, the sugar, and then we are converting it to ethanol. When we are talking about lignocellulosics, that lignocellulosics to ethanol, that process is little bit different from that of the other processes. Besides this ethanol, there are other solvents also which can be produced with the microbiological intervention, that is the acetone and N-butyl alcohol. Some organic acids like citric acid, acetic acid, lactic acid, itaconic acid, and so on. So many acids and alcohols can be produced by the fermentation system. Now, when we are talking about this fermentation system and the product formation, ethanol is one of the most popular fermentation. We can produce glycerol, we can produce acetone butanol, we can produce 2,3-butanediol, and so on. Now, when we are talking about the fermentation with different other products, then it is little bit different, like acetic acid, which can be produced with the intervention of Saccharomyces cerevisiae, Saccharomyces species. Lactic acid, Lactobacillus species are producing lactic acid. Citric acid, Aspergillus niger and other species of Aspergillus are also producing citric acid. Itaconic acid, Aspergillus itaconicus. So, these are some of the example which are well established and well known in the field of biotechnology. Let me give you one particular example of the biologicals which are getting produced inside the microbial cell. Now, when we are talking about this ethanol, I told you that ethanol is one of the biggest fermented product which are available in the global market. Now, carbohydrates are reduced to pyruvate with the aid of nicotinamide adenine dinucleotide, and ethanol is the end product of this particular process. How this particular product is formed? We can use the whole grain, starchy material, sulfide liquid or saccharin material that are used as a feedstock for ethanol production. The organism which is actively participating in this particular conversion is the Saccharomyces cerevisiae, which cannot ferment starchy material directly, but amylases must first break down to starch and then sugar is produced. With this sugar, the fermentation is taking place. Now, when we are talking about such type of product formation, we are talking about C2 product. That means ethanol has got 2 carbon molecules. Now, here if we see the overall production process, now as I have discussed you that ethanol is the product, it can be produced from sugarcane. Now, when it is produced from sugarcane, what we are getting from sugarcane? We are getting sugar, which is sucrose, nothing else. This sucrose is getting converted to ethanol. That means here sugar to ethanol conversion is taking place. Now, when we are going for this reducing that sugar to ethanol, this process is different from that of the process if we are taking any lignocellulosics, that means any biomass, any grassy materials. So, when we are talking about any lignocellulosics, when we are talking about any biomass, then what we are talking about? We are talking about cellulose, hemicellulose, lignin and other minerals, whatever is there present is there inside this reactor system. Now, pretreatment is playing a significant role in this particular conversion type. Now, when we are going, say ultimate product is ethanol. What we are doing is ethanol. Here directly we are converting and we are getting this ethanol, but our source is different. Our source is lignocellulosics. The entire fermentation process is getting changed. Just here is the beauty, here is the freedom, here is the wisdom with the biotechnologists who are designing, who are carrying out the experiment, and that is the reason why biotechnology is so interesting. See, we are going for the pretreatment process. Now, here we are getting the biomasses and biomass is pretreated. How we are going for this pretreatment? How we are doing this pretreatment? We can go for the physical method, we can adopt chemical method, we can adopt biological method. Now, some of these examples are this steam explosion, extrusion, acid alkali treatment, temperature, milling, addition of hot solvent, and if we are going for this biological means we can treat it with the whole cell microbes as a cell to delignify, or we can use some enzymes, that is the enzymatic treatment for removal of lignin from this lignocellulosics. Now, when lignocellulosics, this lignin is removed from this biomass, we are getting available cellulose. This cellulose is undergoing hydrolysis. How we are going for this hydrolysis? We can take any other option, that is either physicochemical process or biological process. That means we can use the hot acid, or we can use some enzymes that may be thermophilic in nature. Multi-enzyme systems can also be used, and the combination of the enzymes and some other techniques can also be taken for hydrolysis of the available cellulosic material to convert this to glucose or the sugar. When sugar is produced, what types of sugar we can expect? We can expect glucose, we can expect xylose, arabinose, uronic acid and so on. And when we are going for, we are getting these sugars, we are going for this fermentation system. Now, what type of fermentation? For ethanol production, it is anaerobic fermentation system. Now, when we are going for this anaerobic fermentation system, we are getting the product which is called alcohol. Now, here this range of fermenting microorganisms are yeast as well as some bacteria. Now, yeast means it is mostly the Saccharomyces cerevisiae. As soon as this alcohol is produced, it is not the pure alcohol, alcohol along with some water molecule. So, we will have to remove this water from this alcohol and ethanol and water. So, alcohol and water mixture is now we are doing distillation, and after distillation we are getting the ethanol, which is having immense application in case of bioenergy or biofuel is concerned, because this biomass to bioethanol, it has got plenty application as per as biofuel is concerned. Now, when we are talking about this is one product, that is ethanol. Now, when I am giving you this ethanol is produced from this fermentation system. Another example I would like to give you is the lactic acid fermentation. This is a type of fermentation which is carried out by lactic acid bacteria, in which sugar, say for example lactose, glucose or any pentose sugars are getting converted either entirely or almost entirely to lactic acid, and when such type of conversions are there, we are calling this type of conversions as homoleptic fermentation, or sometimes we can also go for the mixture of lactic acid and some other product which we are calling it as heterolactic fermentation. In case of homoleptic fermentation, what we are doing, when this glycolytic pathway is used in this lactic acid fermentation, the overall pathway is called the homoleptic fermentation. In it, the pyruvate is directly reduced to lactic acid with the enzyme lactate dehydrogenase. See, this is the pyruvate. In presence of lactate dehydrogenase, it is getting converted to lactic acid. Different microorganisms are participating in this particular conversions are Streptococcus, Enterococcus, Lactococcus, and different strains of Lactobacillus are the microorganism bacteria which are efficiently producing homolactic acid. When we are talking about the heterolactic fermentation, we are talking about this in contrast to homolactic, some microbes carry out this heterolactic fermentation using the phosphoketolase pathway rather than the glycolytic pathway. Now, here see you glucose is converted to glucose 6-phosphate to PPP pathway, see 6-phosphogluconate to ribose 5-phosphate, ribulose 5-phosphate to xylulose 5-phosphate, and one fraction is coming and producing glyceraldehyde 3-phosphate. If you remember PPP pathway, this same pathway is getting followed and ultimately pyruvate is getting converted to lactate. Here some other byproduct, which is this xylulose, when it is coming in contact with this getting converted to acetyl-CoA, acetyl-CoA is getting converted to acetaldehyde and ethanol is produced. So, ultimately in the reaction we are getting the not only this lactic acid, but along with that some amount of ethanol is also getting produced along with the fermentation. That means during fermentation not only the desired targeted product, but some byproduct or contaminated products are also found. So, that is the point of my telling that during fermentation, though we are targeting for the production of single product, but along with this product we may get some contaminants. Say for example, when we are talking about the plant metabolites, yesterday I have already told you that primary metabolites and secondary metabolites. A primary metabolites is an intermediate or product of metabolites which are usually a small molecule that is directly involved in the normal growth, development and reproduction of a particular living cell, whereas secondary metabolites, these are the organic compounds secreted by the plant, are not directly involved in the normal growth, development and reproduction of this organism, but the absence of the secondary metabolite does not result the immediate death, but rather a long term impairment of the organism structure and function. And when we are talking about such type of product formation, then we are calling that yes, along with these particular metabolites, major contaminants are there which are present, because inside the plant cell along with our desired product X, some other products are also present inside this, and this X is not the only one which can be getting extracted when we are removing the cell wall of this particular plant, but it will be whatever is the intracellular metabolites, everything is coming out along with our targeted molecule which is X, and that is the reason for which we are very much interested to remove this contaminant from this cell. So, the production of recombinant biopharmaceuticals can be divided into the following strategies, that is the upstream operation and production and downstream operation. So, if we see now what I have already discussed till now, if I classify the entire lecture, what I have discussed with you, that entire production, that upstream, downstream. Now, if we are talking about this upstream and downstream, we can divide that cell inoculum preparation, media preparation, sterilization, cooling, etc., which are the process are coming under this upstream processing. Then we are going for this fermentation system or the production of metabolites within the particular environment, and we are calling it as fermentation, and this fermentation is the intermediate between the upstream and downstream. As soon as this fermentation is over, the product is produced. Now we will be isolating and purifying this product for subsequent use, and when we are talking about the subsequent purification of our targeted product from the mixture of these contaminants, we are talking about the downstream processing of that product. So, entirely the entire process starting from the manufacture of the metabolites to the purification, if we take this thing, the entire processes, then we can divide it into upstream process and then the transition process where metabolites are getting produced, targeted products are getting produced, which is the transition phase of upstream and downstream, and once the product is getting produced, we start the downstream processing. So, here this fermentation, cell culture, serum collections, whatever may be the system, irrespective of the plant cell, animal cell or microbial cell, when production of product is there, we are talking about the transition phase, and then we are going for the downstream operation. Now, that is the reason why the strategies of biomolecule separation is playing so important role, because this if the targeted molecules are not getting separated with the appropriate concentration with high yield, then we cannot make that process economically viable, and that is the reason why downstream is so important. And while starting this downstream processing, we have to have the idea about the strategies of the downstream processing. Now, when we are talking about the strategies, we are mainly talking about the two processes. If this is a microbial process, we can get this microbial process into two different forms. One is the extracellular metabolite, another is the intracellular metabolite. Now, whatever may be the extracellular, the cell itself, from this cell wall, the product is being secreted to the surrounding medium by this particular cell, and that is the reason why it is extracellular. So, as soon as these metabolites are coming out of the cell, it is getting diluted inside the reactor, and when it is getting diluted, that means dilution problem is much. But though the number of concentrated contaminants are not so high compared to this intracellular, where suppose this is the cell and we are getting our product in the form of the inclusion body, and when we want to get this product, we have to break this cell wall. When we will be breaking the cell wall, whatever may be the thousands and thousands of metabolites are coming out of the system and making the entire contaminants along with our targeted products. So, thousands and thousands of contaminants are there. So, here one problem is the dilution problem, and second problem here the number of contaminants present inside the product. So, what strategy will be taking to select the optimum process for downstream processing? So, in the next class, I will be talking about the strategies of downstream processing. Now, I think I have made you clear that how product is being produced inside the cell, what is upstream, what is transition phase, and what is downstream, where from we can come to this downstream, and problems associated with the downstream processing. Thank you very much.